In Chapter 7, we learn about a wide variety of topics. We're mostly going to be talking about bacteria and their genomes. We're going to talk a little bit about bacterial gene transfer. We're also going to talk about some bacterial defense mechanisms, mostly those that we use for biotechnology purposes. And then we're going to talk a little bit about viruses. Now, because we're covering bacteria and viruses all in one chapter, we really are not going into crazy detail on them, but they are absolutely relevant to our learning because of their technology applications and also because viruses are, of course, an important health risk. We're going to be focusing on bacteria in this chapter, and then of course we'll talk a little bit about viruses. Viruses are non-living particles. We will get to them in just a little bit. Very quickly, we're going to do a review of bacterial cells. Recall that bacteria are prokaryotes. Prokaryotes are single-celled organisms that do not have a nucleus or membrane-bound bacteria. There are two groups of prokaryotes, the bacteria, or eubacteria as they are technically called, meaning true bacteria, and then archaea. We are not going to distinguish between bacteria and archaea for this chapter. In fact, we're actually not even really going to talk about archaea. We're going to focus on bacteria here, especially because we use E. coli as one of our very, very commonly used model organisms. It's sort of our bacteria workhorse. So bacteria come in a variety of different shapes and sizes. Generally, they are round, rod-shaped, or spiral. We could get a little more detailed if we wanted, but that's all we're going to say for now. E. coli are rod-shaped. The term for rod is bacillus. Some bacteria are photosynthetic, so of course that means they're very important in ecosystems, particularly aquatic ecosystems, for fixing carbon, converting it into usable energy. And then there is a whole variety of other metabolic types for these organisms. Replication occurs for preparation for cell division. Cell division in bacteria is called binary fission. It is roughly similar to mitosis. It's not exactly the same, but there are some similar elements that suggest that mitosis evolved out of the binary fission mechanism. But you can kind of think of it as similar, roughly similar to mitosis, since we're not going into all of the details there. So of course, E. coli is our model genetic organism. This doesn't mean that we don't use other bacteria, but E. coli has been extensively studied for a very long time. There are all kinds of different mutants of E. coli, and there are a variety of different natural strains of E. coli. The reason that E. coli was initially used as a model organism is that it was really easy to acquire. Basically, most, or at least many individuals, humans included, many mammals have E. coli in their intestinal tract. So it's a organism that lives in the gut of warm-blooded mammals. And so when there were not companies that you could buy these organisms from, you had to collect them themselves. And so these were usually collected from feces of warm-blooded animals. The genome is relatively small because it's a bacteria. It's only got about 4,300 genes or so, 4.6 million base pairs. We haven't talked about the human genome yet, but human genome is much, much bigger. It's got about 22,000 genes located in the genome. So you can see that it is really, really large. This image here shows an E. coli cell that has burst, and this is its genomic DNA that's sort of spread out around it. There's some stats here for the bacterium. You may have a handout of this, or it's posted in Brightspace with just some general information and how we use E. coli. And then there's also a quick example of their reproductive mechanism, binary fission, basically cloning. And then there's also a diagram here of conjugation, which we'll talk about in just a few minutes. So why do we use E. coli? Well, there's a couple reasons that E. coli are a great model organism. One, it's incredibly tiny. This counts for all bacteria. But this one in particular is very small, and it's very easy to grow. When it's growing and it's very happy, it divides about every 20 minutes. So really, really fast reproduction. This means we can double the E. coli population every 20 minutes. So we can start out with a very, very small number of cells and increase them rapidly. This is also really great if we're using it for biotech purposes because it just makes our experiments go that much quicker. You can grow these guys on either a liquid medium, we would call that a broth, with all of the nutrients in it that the bacterium need. You can also grow it on a Petri plate. We would call this agar. Again, the nutrients, just like in the broth, are in the agar, and the bacteria grow on that agar and bring in the nutrients. There are many mutants that are commercially available, so you can do a lot of things with this guy really easily. The genome has been mapped for quite a while, so you know where all the restriction sites are, you know where all the genes are, all the genes that they are, all of the different types of genes, you know what all of the genes do, or at least most of them, and so it's very easy to use this organism for genetic engineering. So I'm just going to let you pause here and write all of this down. This is just a laundry list of the reasons that we like using bacteria and viruses for genetic studies. I'm not going to read them because I don't have too much to elaborate on, but go ahead and pause and get these down in your notes. So same thing here. I'm going to ask that you just pause the video, write down E. coli's contributions. These are also on the E. coli handout. So if you have that handout or want to print it up from Brightspace, you don't need to write this all down. If you don't want to do that, just go ahead and jot these notes down really quickly. This image here shows an electron microscope image of E. coli. You can see that rod-shaped bacteria. These guys tend to stain a kind of a pink color. You can see that on the previous slide. This is based on their cell wall characteristics. And we call these guys a gram-negative bacteria. Gram is a type of stain. They don't hold the stain well, and so they look pink instead of purple, which is the gram color. Most of our bacteria are going to have a singular circular piece of DNA. They tend to have only one chromosome in their cell, although there are some odd examples that where bacteria have more than one chromosome or linear chromosomes. This is relatively rare. There's only a few examples that we know of both of those. Our genome is going to contain or our genomic chromosome is going to contain all of the essential genes for survival. So all of the genes that are required for that organism to sort of perform its daily tasks, metabolize its food, communicate with other organisms in its colony, all of that is going to be contained in the genomic chromosome. And then of course all of the instructions for replication and all of that as well. Bacteria may also have plasmids. These are little tiny circular chromosomes that are separate from the genomic chromosome. They replicate separately and have a different set of genes than that genomic chromosome. So plasmids are essentially extra DNA. They're very, very small compared to the genomic DNA. Generally, they contain non-essential genes, but they may contain genes that are going to be required for survival in a particular environment. So, for example, there are antibiotic resistance plasmids that contain genes to metabolize antibiotics so they can survive in the presence of antibiotics. There are also plasmids that are important in gene transfer. We'll see one of those in a later video. And plasmids have been sort of commandeered by scientists for the purpose of genetic engineering. Plasmids are small and they're easy to transfer between bacteria. In fact, bacteria do it on their own sometimes, and we're going to talk about some of the mechanisms by which bacteria uptake or transfer genes between one another. And we have used that to our advantage. Because plasmids are so much smaller than the bacterial genome, they're really easy to get in and out of the cell. They have only a few genes, and so they're very easy to engineer with a gene that we might want the organism to have. This image here is not to scale. It's a little bit misleading. It's actually much, much smaller than that genome. There are some specific important components of a plasmid. Plasmids are always going to have restriction sites, and you need those restriction sites to be able to cut the plasmid open, to cut the gene that you want out of the genome and insert it in with those sticky ends. And we've probably talked about this in lab already, but we'll talk about it again later in this chapter. Plasmids always have an origin of replication. This is for DNA replication. And then they usually have some sort of selectable marker so that you know that your cell has a particular plasmid that you want. Very often they have an antibiotic resistance gene so that they can survive in the presence of an antibiotic, whereas other bacterial cells cannot. So this is going to reduce contamination, and it's going to reduce the number of non-plasmid bearing cells. And then we usually have some sort of selectable marker, most likely in lab. Oh, nope, not yet. Sorry, wrong class. I was thinking about the wrong class. Most likely they're going to have some sort of selectable marker that's going to help you know that you have a plasmid with that inserted gene. We will talk about this a little bit later in our semester. And then we've got some other regulation sites here. We've got promoters. This is to turn the gene on to make sure that your sequence is expressed, so that inserted gene gets expressed. And then we've got these primer sites. That is for replicating the gene of interest. We'll talk about that a little bit later. But these plasmids are going to contain, the engineered plasmids anyway, are going to contain these specific sites. In normal plasmids, natural plasmids, they're just going to contain the genes that they're carrying, that they're being selected for, and then that origin of replication. A third type of piece of genetic information is called an episome. An episome is a plasmid that can integrate into the chromosome. They can actually pop out, which is kind of crazy, but they replicate separately from the other plasmids and from the chromosome, and are sort of unique in their ability to pop in and out of the bacterial genome.